Hi everyone, let's explore what oncogenes are and their role in cancer. To understand oncogenes, first we must understand the cell cycle. The cell cycle controls the process of cell division. A cell holds DNA, which acts as its brain due to the specialized pattern of the DNA's gene expression. This results in memory, so the cell can remember a specific function to perform. Cells receive signals from their environment, other cells, and growth factors and enter into the cell cycle when required. There are 4 phases in the cell cycle: G1, synthesis, G2, and mitosis. Throughout the cell cycle, there are checkpoints for detecting damages, alterations, or mutations of cells. Mutations means changes in genes. If any damage is identified, specific genes called tumor suppressor genes activate and repair the damage. Cell growth and division in the cell cycle are controlled by various proteins. Proteins are produced by genes called proto-oncogenes. They are healthy, beneficial genes and produce a protein involved in cell growth, division, and differentiation. They are typically only turned on when received a signal to grow. Oncogenes are mutated dysfunctional proto-oncogenes. Oncogenes cause excessive cell division and uncontrolled cell proliferation. Oncogenes are dominant genes. They are able to be activated with a single event, such as a point mutation or UV light. One mutated copy is enough to trigger tumor formation. However, oncogenes exclusively do not cause cancer. Tumor suppressor genes are responsible for preventing mutations by inhibiting proliferation and division of damaged cells, repairing that damaged DNA, and sending signals to initiate cell death, called apoptosis. TSGs are a recessive gene. We get one copy of the TSG from each parent. When both copies of TSGs are mutated, the chances of developing cancer increases. Mutated TSGs are unable to fulfill their roles and will activate oncogenes. Mutations can occur from point mutations, chromosome translocation, or amplification. Along with activated oncogenes, we must have the inactivation or mutation of both copies of TSGs to cause uncontrolled cell proliferation. Now that we understand the relationship between oncogenes, proto-oncogenes, and TSGs, let's put this relationship into perspective by using the RAS oncogene as an example. A cell receives a signal from the epidermal growth factor, which binds to and stimulates its receptor, the EGFR. This results in the activation of the RAS protein, which signals the activation of something called a mitogen-activated protein kinase pathway. The MAPK pathway is essentially the on button for other proteins that are responsible for cell proliferation or death. Normally, it can be switched back off. When the RAS protein is mutated, this results in the continuous signaling of the MAPK pathway, resulting in the overproduction of genes and proteins, which means an uncontrolled overproduction of daughter cells. Thus, this increases the chances of cancer formation. In conclusion, we've covered the critical role that oncogenes play in cancer formation, and I know it's scary to think that just one gene mutation can cause cancer, but the unfortunate truth is that oncogenes do have the capacity to cause rapid, dysregulated cell growth, which leads to cancer or tumor formation. Thank you, and here are your key terms.